The history sounded certain. The DNA did not. There's one genetic fact that makes the traditional origin story of an ancient Jewish population incomplete. Not wrong, not fabricated, just incomplete in a way that changes everything. For generations, the narrative felt stable. Roots in the Levant, dispersion through exile, preservation of identity across continents. A straight line through time. A people carrying memory, ritual, language, and law from one geographic point outward. But when researchers began comparing modern genomes with ancient DNA samples, the line split. One branch pointed clearly back to the Near East. The other did not. And that difference is not technical. It is structural. Because when two genetic lines tell two slightly different stories, the question is no longer, where did they come from? It becomes, what actually happened? In this video, we are not going to argue myths. We are not going to chase ideological interpretations. We're going to do something much simpler and much more revealing. We will assemble the evidence the way geneticists do. The paternal line, the maternal line, population comparisons, migration layers, and we will look at what the data actually allows us to conclude and what it does not. Because here is the part that unsettled researchers most. The paternal lineage, passed from father to son, shows a strong, consistent connection to the Levant. The genetic markers cluster near populations, historically rooted in the Eastern Mediterranean. That part aligns with long-held historical memory. But the maternal lineage tells a more complex story. In multiple subgroups, mitochondrial DNA, inherited only through mothers, reveals lineages that appear deeply European in origin. Not recent, not modern, ancient. That is not a contradiction. It is a trace of events that do not appear clearly in written records. And once you see it, the simple version of the story does not collapse, it expands. But before we go further, we need to understand the tools that made this possible. Because this is not guesswork. It is not speculative ancestry testing. It is population genetics built on three very specific instruments: the Y-chromosome, mitochondrial DNA, and ancient DNA extracted directly from archaeological remains. Think of them not as abstract lab terms, but as investigative devices. The Y-chromosome is the surname of the genome. It passes down almost unchanged from father to son. It forms clusters across geography and time. When you compare Y markers across populations, patterns emerge, patterns that often trace back to ancient regions. Mitochondrial DNA is different. It moves through mothers. It survives bottlenecks. It preserves small founder events. It can reveal when a limited number of women shape the future of an entire population. And ancient DNA changes the scale entirely. Because instead of comparing modern populations and guessing backward, researchers can now compare living genomes to actual remains from 2,000 years ago, sometimes more. No mythology, no romanticism, just statistical clustering, mutation rates, and geographic anchors. That does not make the story cold. It makes it measurable. And here are the rules we will follow. No sweeping claims. No cultural judgments. No identity politics. Only what comparative data supports. If a lineage clusters near Levantine populations, we say so. If another clusters near early European groups, we say that too. If something remains uncertain, we leave it uncertain. Because the goal is not to simplify identity. It is to understand it. Now here is the tension. If you look only at the paternal line, the story feels almost complete. A Near Eastern origin, migration into Europe, continuity across centuries. The markers align. The clusters are visible. It gives you something solid. But the maternal line complicates that stability. And complication is not chaos. It is evidence of history unfolding in ways that written sources rarely capture. How did paternal markers remain strongly Near Eastern, while maternal markers diversified in Europe? Was it gradual integration? Localized founder effects? Population bottlenecks? Social structure shaping marriage patterns? The data does not shout. It suggests. And what it suggests forces us to reconsider how identity forms over centuries. Because identity is not a single strand. It is braided. And somewhere in that braid is a moment, or many moments, when history moved differently than memory alone records. We will begin with the paternal line. It appears straightforward, almost reassuringly so. But that clarity will not hold for long. We are not looking for legends. We are looking for signals in DNA. And the clearest signal comes from the Y chromosome. Unlike most of the genome, the Y chromosome changes slowly and travels almost untouched from father to son. Over generations, it accumulates small mutations, markers. Those markers form branches. Branches form clusters. And clusters tend to anchor themselves to geography. When researchers mapped the Y chromosome markers of this ancient Jewish population and compared them to surrounding populations, something stood out immediately. The strongest clustering pointed toward the Levant. Not vaguely, not symbolically, statistically. When plotted against large reference datasets from the Eastern Mediterranean, populations historically rooted in regions like modern-day Israel, Lebanon, and parts of Syria, the paternal markers consistently fell within that orbit. That matters, because genetic clustering is not based on language or religion. It is based on shared mutations inherited over thousands of years. When two populations share specific Y chromosome haplogroups at elevated frequencies, it usually reflects a common ancestral pool at some point in the past. In this case, the paternal signature did not scatter randomly across Europe. It did not drift primarily toward Central or Northern European clusters. It stayed tightly connected to Near Eastern lineages. Visually, the pattern is clean. Imagine a map. Markers lighting up across Europe, but the densest overlap, the deepest root, pulling back toward the Eastern Mediterranean. That becomes the anchor. And here is where the investigation becomes more precise. Researchers do not simply look at modern comparisons. They also compare these Y markers to ancient DNA samples recovered from archaeological remains in the Levant dated to the Iron Age and earlier periods. When overlaps appear between modern paternal lineages and ancient Levantine samples, the signal strengthens. It suggests continuity, not uninterrupted isolation, not genetic purity, continuity. This does not mean nothing changed over centuries. It means that the paternal core retained a detectable connection to a Near Eastern origin. If we stopped here, the story would feel almost settled. A Near Eastern founding population, migration into Europe during antiquity, cultural preservation reinforcing internal continuity. The genetic data would align neatly with historical memory, and to a large extent it does. But let's stay in this layer a little longer, because even anchors have texture. Within the paternal line, certain haplogroups appear at higher frequencies than in surrounding European populations. These lineages are not exclusive. No haplogroup belongs to one group alone, but their concentration patterns matter. In comparative studies, these Y lineages show closer affinity to populations historically situated in the Levant than to populations in Western Europe. That suggests that when this ancient Jewish community settled in Europe, a significant portion of its male ancestors carried Near Eastern paternal markers, and those markers remained relatively stable across generations. Why would they remain stable? Population structure. Historical records indicate that in many regions, Jewish communities practiced endogamy, marriage within the community, often shaped by social boundaries, religious law, and external restrictions. Endogamy does not freeze genetics, but it slows dilution. Over centuries, that can preserve specific paternal clusters more clearly than in populations with constant large-scale intermarriage. So the paternal line becomes both a genetic signal and a historical reflection. Now here is the important part. The Y chromosome tells us about male ancestry lines. Only male lines. If one founding population consisted primarily of men migrating into new regions, and if they formed communities that later incorporated local women, the Y chromosome would preserve the geographic memory of those men more strongly than other parts of the genome. That possibility is not speculation. It is a known pattern in many migratory histories worldwide. But for now, the paternal evidence gives us something firm. A geographic anchor in the Levant, a statistically significant clustering, a line that appears consistent with historical narratives of Near Eastern origin. If this were the only dataset available, researchers would likely describe the origin story as largely confirmed. The data feels coherent. It feels stable. It feels almost finished. And that feeling is exactly why the next layer matters. Because when the same researchers turned to mitochondrial DNA, the maternal line, the pattern shifted. Not slightly. Structurally. If the paternal line provides an anchor, the maternal line introduces movement. And once we add that movement, the story stops being linear. If we looked only at the Y-chromosome, we might conclude the investigation here. The origin appears Near Eastern. The genetic trace aligns with tradition. The cluster is visible. But that clarity depends on looking through one lens. The moment we switch lenses, the image changes. And in the next block, we will examine the maternal line, and the moment when two true genetic signals begin to diverge. Now we turn to mitochondrial DNA. And this is where the map shifts. Mitochondrial DNA does not travel through fathers. It passes from mothers to their children, sons and daughters. But only daughters continue transmitting it. Like the Y-chromosome, it changes slowly over time. It forms lineages. It forms clusters. But when researchers mapped the maternal lineages of this ancient Jewish population, the geographic signal did not mirror the paternal one. Instead of clustering tightly around the Levant, several major mitochondrial lineages pointed toward Europe. Not recent Europe. Not modern assimilation. Deep European roots. This is the moment where two truths collide. The paternal line shows strong Near Eastern continuity. The maternal line, in multiple subgroups, shows substantial European ancestry. At first glance, that sounds contradictory. It is not. It is historical. When population geneticists examined the most common mitochondrial haplogroups within this community, they found that a small number of maternal lineages accounted for a large proportion of individuals. This is known as a founder effect, when a limited number of ancestors contribute disproportionately to future generations. And when those maternal haplogroups were compared to large reference databases, their closest affinities often aligned with ancient European populations. Southern Europe, parts of Central Europe. In some cases, lineages tracing back to prehistoric European maternal branches. That does not erase Levantine ancestry. It complicates it. The simplest explanation, and the one most supported by demographic modeling, is that early male migrants from the Near East established communities in Europe and, over time, incorporated local women. Those women became ancestors of entire maternal lines that persist today. This is not unusual in human history. Migration is rarely symmetrical. In many historical expansions, whether trade-based, forced, voluntary, or gradual, male-driven migration patterns left a stronger paternal geographic signal, while maternal lineages reflected local integration. But here's why this matters. Cultural continuity can remain strong even when genetic inputs diversify. If a community preserves language, law, ritual, and internal structure, it can maintain identity across centuries, even as its maternal ancestry becomes partially local. This does not weaken the story. It makes it more human, because identity is not a fossil. It is a living system. Now consider the scale of this effect. In certain subgroups within European Jewish populations, for major mitochondrial lineages account for a large percentage of maternal ancestry. Genetic modeling suggests that some of these lineages expanded dramatically during the medieval period, possibly after population bottlenecks. Bottlenecks compress diversity. When a population shrinks due to migration pressures, persecution, economic restriction, or geographic isolation, a small number of surviving lineages can shape the genetic future of millions. This is why founder effects matter. And this is also where genetics intersects with medical research. Certain inherited disorders appear at elevated frequencies in specific Jewish populations, not because of identity, but because of historical isolation combined with founder effects. A small number of ancestral variants became amplified over generations. Understanding these patterns has been crucial for medical screening programs and preventive health care. But the medical dimension is a consequence, not the core story. The core story is this. The paternal line preserves a Near Eastern anchor. The maternal line reveals European integration. Two true signals, not rivals, layers. Now step back and imagine the historical reality behind those signals. Communities forming along trade routes. Families establishing roots in Roman Europe. Local women joining tightly structured religious communities. Children inheriting language, law, and memory from fathers, and mitochondrial DNA from mothers whose ancestors may have lived in Europe for thousands of years. No single written source captures that complexity fully, but the genome does. And here is where a powerful metaphor helps. Think of language. Take Yiddish, historically spoken by many European Jewish communities. It blends High German structure with Hebrew vocabulary and Slavic influences. It is not purely one thing. It is layered. Genetic ancestry can function the same way. A cultural framework anchored in the Near East. A maternal contribution shaped in Europe. A population history that braided both strands into something distinct. It is not dilution. It is formation. Now remember the earlier question. How did paternal markers remain strongly Near Eastern while maternal markers diversified? Social structure. Marriage networks. Religious law tracing lineage through mothers for identity, but not restricting ancestry to one geography at the beginning. Over centuries, the combination of endogamy and founder effects stabilized the mixture into something recognizable and persistent. And this brings us to the next tension. If this pattern appears clearly in European branches of this population, what happens when we examine communities outside Europe? Communities in Ethiopia, in India, in parts of Asia, do their genetic patterns follow the same paternal-maternal structure? Or does geography reshape the mosaic again? Because if identity can survive maternal integration in Europe while preserving Near Eastern paternal continuity, what happens when the geographic context changes entirely? That is where the next layer of the investigation begins. And it may be even more complex than the European case. If the European case shows us a genetic mosaic, what happens when geography shifts even further? Let's step outside Europe. Jewish communities formed in places far from the Mediterranean world, in Ethiopia, in India, in parts of Central and East Asia. These communities preserved religious practice, ritual law, collective memory, and a shared sense of belonging. But when geneticists examined their DNA, the patterns did not always mirror the European model. In some of these groups, paternal markers still show varying degrees of Near Eastern connection, sometimes strong, sometimes partial. In others, the signal becomes more regionally blended. Maternal lineages, in particular, often reflect substantial local ancestry. This is not evidence of discontinuity. It is evidence of adaptation. When a community settles in a region for centuries, sometimes more than a millennium, it inevitably interacts with its surroundings. Geography matters. Demography matters. Social structure matters. The longer the timeline, the more the genome reflects both origin and environment. Take the Ethiopian Jewish community as an example. Genetic studies suggest a complex picture, one that includes local East African ancestry alongside signals that may trace back to broader Near Eastern lineages. The Indian Jewish communities show yet another pattern, significant South Asian genetic components, combined with varying degrees of West Eurasian ancestry. In each case, identity endured, not because DNA remained unchanged, but because identity is not determined by a single chromosome. Here is the key shift. Genetics can tell us where ancestors came from. It cannot fully define who a community becomes. Across continents, Jewish communities maintained shared religious frameworks, textual traditions, dietary laws, liturgical language, and collective memory. Those elements created continuity even as genetic inputs diversified. In some cases, the genetic profile of a community becomes deeply local, reflecting centuries of residence and integration. In others, historical isolation preserves clearer ancestral signals. There's no single pattern that applies everywhere. And that is precisely the point. The search for a pure, linear origin collapses under real data. What emerges instead is a mosaic, a paternal anchor pointing to the Levant, maternal lines revealing integration across Europe, regional adaptations shaping communities in Africa and Asia, different combinations, different proportions, one evolving identity. If we reduce identity to DNA alone, we lose history. If we ignore DNA entirely, we miss hidden chapters of history. The truth sits in between. Genetics shows movement. History records memory. Culture preserves structure. Together, they form continuity. Now return to the question from the beginning. Why did one genetic line lead clearly back to the Levant, while another told a more regionally blended story? Because populations are not static. Because migration is rarely symmetrical. Because communities form through both preservation and incorporation. The paternal line gave us an anchor. The maternal line revealed integration. The global cases showed adaptation. And when we assemble those layers, a deeper pattern appears. Identity is not a straight line. It is a braided one. It contains origin and journey at the same time. This does not weaken ancestral connection. It contextualizes it. An ancient Jewish population can carry detectable Near Eastern paternal ancestry, significant European maternal ancestry, and still remain culturally and historically continuous across centuries. That continuity does not depend on genetic uniformity. It depends on shared structure and memory. History is written in books. It is also written in DNA. But neither replaces the other. The genome reveals where people moved. Tradition reveals what they carried with them. And when researchers uncovered these surprising DNA origins, they did not dismantle the past. They refined it. They showed that the question, where do we come from, rarely has a single geographic answer. It has layers. If you want to explore another DNA story that challenges the simple version of origin, subscribe and stay with the investigation.